All right, so this is the continuation of the chapter 6.2, the viral life cycles. So this is part 2. We left off with our prokaryotic viruses, and now what we're going to talk about are the life cycle of viruses with animal hosts or eukaryotic cells. So we'll start with animal hosts. And the way that we start is basically like the lytic cycle. So it follows similar steps as the lytic cycle that we saw with bacteriophages. So step one is attachment. And this is very similar, where we are taking the virus and it is going to attach to the outside of the cell. Of course, we're not talking about the cell wall, because when we're talking about animal cell, which I should write here, so for animal cells, we don't have a cell wall. So when we're talking about attachment, it's attaching to the plasma membrane. And so this is similar to the lytic cycle with the bacteriophage. Where we change things up a little bit is here in step 2. So it's still penetration. However, that is going to occur differently. So we don't have the same structure to the virus as a bacteriophage. Remember, bacteriophage is very specific. With our animal hosts or our eukaryotic viruses, we have all those different types of styles of viruses. So they could be helical, they could be dodecahedron shaped, they could be various different shapes. They're not that very distinct bacteriophage shape that we saw with the capsid and the sheath and the tail fibers. So when we talk about the viral life cycles with animal cells, what we can say is that in penetration, it's going to enter the cell, and there are two different ways. It's going to be either by endocytosis or by membrane fusion. So I am going to move this over so that we can talk more about this. So endocytosis, in this case, what we're talking about is engulfment of the virus. Engulfment of the virus. And then when we talk about membrane fusion, this is where the viral envelope is going to merge with the host cell membrane. So the viral envelope is going to, I'll say, merge with the cell membrane. So if we think about this, right, when we're talking about a eukaryotic cell, right, so then we have our nucleus, because when we're talking about a eukaryotic cell, we're talking about a nucleus here. Then we have our virus that comes along. So we have our virus. When we talk about a virus, we're going to say that this one has an envelope. So it has an envelope on the outside there. Actually, let's say that's the envelope. And then inside here is where we have our virus. And then inside the virus is where we have the viral nuclear information. So this right here is the envelope of the virus. This right here is the plasma membrane of the eukaryotic cell or the animal cell. So when we're talking about entering the cell via endocytosis, we can bring this into the cell. Let's say we'll take a look at both types of viruses here. So this is our non-enveloped virus, right? So there's no envelope here, and then this one is enveloped, so there is an envelope. So here, let's talk about endocytosis. So in this case, remember, we have engulfment of the virus. So in this case, we have attachment here of this virus, and then the plasma membrane, let's erase this. The plasma membrane is going to come up and it's going to engulf. So endocytosis, we kind of have these pseudopods that are going to come up of the plasma membrane, and it's going to engulf or grab itself around this virus. It's going to keep doing that until these two parts merge. And then what we have is this virus that is now going to come into the cell. And so if we take a look here, I should really make that smaller. What we have done is now we have a virus that's come into the cell, and then we still have our plasma membrane out here. And so this is going to come inside of the cell, and then it's going to go through the next steps that we'll talk about in just a moment. Now, if we talk about through membrane fusion, this is when we have our enveloped virus, which we have here. And our enveloped virus is where we're going to merge that viral envelope with the cell membrane. So here we have attachment, and then what's going to happen is this membrane here, this envelope, is going to just merge with our plasma membrane of the cell. So we have this, it's going to merge with it, and then as it's merging, then what we have is something that looks like this. And then, of course, this is moving into the cell. So then what we have is this plasma membrane, and now we have a virus that's inside of the cell. And so now we can see that this virus, this first one that came in through endocytosis, now inside of the cell has a plasma membrane. This one, the second one that came in through membrane fusion, does not have a membrane. So once it gets in, it's just going to be a naked virus with the nuclear information over here. Now we have this one that has the plasma membrane because it grabbed that plasma membrane as it came in. So then when we talk about that in step 3, this is when we have uncoating of the virus in the cell. Uncoating of virus in cell. So this is where we have these viral contents are going to be released to the inside of the cell. So in here, in this first one we're talking about is we're going to be breaking apart this kind of newly formed plasma membrane. There are various ways that this happens. We're not going to get into those details. But then this is going to allow the virus particles to come out of here. And then it's going to separate even further. It's going to uncoat, and separate these capsid pieces, capsomeres, and then release the nuclear information, whether it's RNA or DNA, into the cell, and then we'll move on to the steps depending on what it is there. Same thing with this other one here. We're going to be breaking it apart and then releasing the contents into the cell. So we're uncoating of the virus, releasing all of the particles inside of the cell. But keep in mind, as we're doing this, we haven't harmed the cell in any way. So it's come into the cell, right? So the virus has come into the cell either through endocytosis, which didn't hurt the cell, or membrane fusion, which didn't hurt the cell. So the cell is still kind of none the wiser. It's going to be uncoating the virus inside the cell. Then we have step 4. In step 4, we have biosynthesis. And in biosynthesis, we have the nuclear information is then going to start to make copies, right? So just like we saw with the bacteriophage, we can see our viral nucleic information makes copies. It's actually, instead of saying makes, we're going to say hijacks machinery. And we'll talk about the different ways that the different types of nuclear information do that. So some of them are going to enter the nucleus, and it's going to be replicated there. And then it's going to make all of its different parts through transcription and translation. But we're going to talk about those specifics in just a moment. So in step 4, we have biosynthesis. And then in step 5 is when we have assembly. So assembly, again, just like we saw with the bacteriophage, this is when our new viral particles are going to be assembled. All right, so if we're kind of following along with these parts, we have all of our kind of pieces are all out here in biosynthesis. And then in our assembly, now we're just piecing these all together. So we're going to be putting these all together here, putting these all together here, and then we're putting the nucleic information in there. And now we've assembled our viruses. Then we have step 6. Oops. And then in step 6, this is when we have release. And in release, this is when our new viral particles are exiting the cell. So new viral particles exit the cell. Now, in this case, when these new viral particles are exiting the cell, the host cell is going to continue to make viral particles. So the host cell continues to make viral particles. And this is because as these exit the cell, they actually aren't hurting the cell. So we see this through exocytosis, right? So this virus, this newly made virus, this virion, is going to come up to the plasma membrane, and then it's going to bud out, is what it's called. So bud out, or budding, is the process of this virus leaving the cell. So it's going to kind of go out in this way, and then it's going to pinch off as it has gone out. And then what the cell is left with is just its regular plasma membrane, and then now this goes out and it has a nuclear envelope. And so then this one is no more because it just left. And so then we can see right here that this cell is just fine. And what it's doing then is it's going through and it's making more particles. So it's going through the same steps, steps 4, 5, and 6 over and over and over again, because these things are budding from the cell, and it's not harming the cell, it's just taking some of the plasma membrane with it. But we might have other ones that are coming in and bringing more plasma membrane with it anyway, which is adding to the plasma membrane. So then this cell can continue making viral particles. And this is an important distinction between the bacteriophage, which in the lysogenic, or in the lytic cycle, it actually has to kill the cell. It bursts the cell, it lyses the cell in order for it to escape the cell. We don't see that same situation here with animal cells. All right, so I kind of want to go back to, not kind of want to, I'm going to go back to the different types of nucleic information. And this is important because we see different types of nucleic information depending on the type of virus it is. And so as we're moving through the chapter and you are learning about the different types of viruses, it's important to understand what type of virus it is. So animal viruses can be DS DNA. So that's double-stranded DNA, and we've already spoken about this in a previous chapter, but we're going to get into more details now. So if the virus has double-stranded DNA, or dsDNA, this means that it can be directly replicated by the host. Right, so if it has double-stranded DNA, that's just like the chromosome in the host cell, which means that that double-stranded DNA of the virus, once it gets inside of the cell, can go right into the nucleus. It goes into the nucleus and can just get itself replicated and copied over and over and over again, right along with the nucleus of the cell. So that can be directly replicated by the host. There's also single-stranded DNA. And when there's single-stranded DNA, then we can just use the host machinery. Then the host makes it double-stranded DNA. Then it will replicate it, just like double-stranded DNA. So if a piece of single-stranded DNA is found inside of the cell, just like if we found it inside of any one of our animal cells, if it finds single-stranded DNA, it's just going to make the other strand, and then it's going to incorporate it and replicate it. Now, if we have a RNA virus, we can have 3 different types of RNA viruses. We can have double-stranded RNA. We can have single-stranded RNA, but it's a positive single strand, so positive single-strand RNA. Or we can have a negative single-strand RNA. So we have different ways that we're able to utilize the RNA viruses. So if it is a positive single-stranded RNA, then this can be translated directly. So similar to double-stranded DNA that can be directly replicated, our positive single-strand RNA can be translated directly to proteins by the ribosomes. And that is because it's like mRNA. So our cells, animal cells, see the positive single-strand RNA. So positive single-strand RNA is the correct strand. It's the strand that contains the information to make a particular protein. So in our bodies, when we make mRNA, which you should be very familiar with, when we make mRNA, that's a positive single-stranded RNA. So that contains the information for a protein. So if a virus comes in and it contains for its nucleic acid or for its nucleic information a positive single-stranded RNA, then it can just, our cells can just use that just like it's mRNA because it sees it as mRNA. So it can be translated directly to a protein. So it can get into the cell, and that information from that virus can be translated directly into making all of its capsomeres and all of the information that it needs, all of the pieces that virus needs to then exit the cell. Now, if it comes in with a negative single-strand RNA, then it's first replicated into a positive single-strand RNA. And it does this by RNA-dependent RNA polymerase. That's the enzyme name, shortened to RDRP. So RNA-dependent RNA polymerase. RNA-dependent RNA polymerase actually comes in with the virus. So the virus itself, if it's a negative single-strand RNA, it's going to also have RNA-dependent RNA polymerase, or RDRP. And if it has RDRP, then what it's going to do is it's going to utilize RDRP to make the positive single-strand RNA. So that then the positive single-strand RNA can be translated by the ribosome directly into a protein, and then it can make copies of itself. Now, I skipped over the double-stranded RNA, and this is because the double-stranded RNA has the positive strand and the negative single strand. So it can take the positive single strand, so the positive single strand RNA is going to be translated directly. So it's going to separate it, and this one is going to be translated directly. The negative single strand RNA is going to use the RDRP to make the positive single strand DNA to then be translated. So we see both things happening with the double-stranded RNA. And actually your text has a really nice flow chart. So typically we see, DNA goes to RNA, which goes to protein, right? Which is kind of what we see in our typical eukaryotic cells or animal cells. We have our DNA, we make copies of it, or not copies, but we transcribe it into RNA, and then we translate it into a protein. We can see this with viruses. Remember up here, we have our DNA viruses, but if we have RNA viruses that we're infected with, we have a different situation. And so if in the text it has something that looks like this, so positive single-strand RNA can automatically be translated into the viral proteins. If we have a negative single-strand RNA, then first it's going to need to be turned into a positive single-strand RNA, and we do that using our DRP. So our DRP can change it into a positive single-strand RNA, and then we can make our viral proteins. But if we have our positive single-strand DNA, we can also use our DRP to make a negative single-strand RNA. So we have two copies there. It also kind of protects the positive single-strand RNA. We'll talk about that later. But we can make we can utilize our DRP to make the negative single-strand RNA, which can then be made again with our DRP to make a positive single-strand RNA to make the viral proteins. Right here, this is the host ribosomes. Now, something else that you can think of in here is if we have double-stranded RNA, then we can use our DRP to make a positive single-strand RNA with that negative single-strand RNA. And then we can again go to our viral proteins through the host ribosome. So a little bit different flow of information here compared to the DNA goes to RNA, goes to protein. Now, an exception to these rules are what are called retroviruses. Now, retroviruses are positive single-strand RNA viruses that has a viral gene for reverse transcriptase. So a viral gene, meaning that it brings it with it for reverse transcriptase. ACE meaning enzyme. So if you look at the name of this enzyme, reverse, so going backwards, right, in reverse, transcribing ACE. So it's an enzyme that is going to reverse transcribe. What this means is that we're taking our positive single-stranded RNA, which comes in with retroviruses, and then we're going to use reverse transcriptase to make our single-stranded DNA. So reverse transcriptase to make our single-stranded DNA. Then we can make our double-stranded DNA just using our typical machinery inside of our eukaryotic cell. Then that double-stranded DNA is going to be integrated into the host genome. When it's integrated into the host genome, this is called a provirus. So let me write that definition down here. So provirus is when viral DNA is integrated into host DNA. But when we're talking about it now, we're talking about it in an animal host. So this is our viral DNA is integrated into host DNA. So recall that we mentioned a prophage. So remember, a prophage is a phage that has integrated its genome into the host chromosome. In this case, the provirus is a eukaryotic virus that has integrated its genome into the host genome or the host DNA. Unlike a prophage, though, proviruses do not undergo excision. So I'm going to write that down. So unlike a prophage, a provirus does not undergo excision. So a provirus never gets cut out. A provirus is kind of similar to the lysogenic cycle in that once a retrovirus has taken its positive single-stranded RNA, it has used reverse transcriptase to turn it into single-stranded DNA. Then that turns it into double-stranded DNA, just utilizing the mechanisms of the cell. Then it can use the enzyme integrase. We've mentioned it before, integrase, to integrate itself into the host genome. So that's up here. Integrase is used to integrate it into the host genome. Now it's a provirus. That provirus stays in there all of the time. And so what it does is it stays in there, and as it stays in there, when that eukaryotic cell is replicating its DNA and making new copies of the cell, that virus goes with it. As that cell is existing, it's going in and it's making copy after copy after copy. It's transcribing, it's translating all of the information for that virus. And that's why things like HIV, which is a retrovirus, are very, very dangerous. So we're going to talk a little bit more about the difference between a persistent infection and a latent infection, a chronic infection. So let's talk about that now. So persistent infection. A persistent infection is when a virus stays in a certain tissue or in an organ of the infected person. So when a virus stays in a certain tissue or tissues or organs of an infected person. So persistent, meaning it continues on. So it's constantly infected. So this is when the body didn't get rid of all of the virus. So it means that it has become a provirus most often, and that's when it's going to stay inside of a particular tissue or an organ or a system, and then it's going to stay in there. So when it does stay in there, the virus can be silent or it can be productive but without harming or killing the host. So it can be silent or productive without harming or killing the host. And then it can do this by either regulation of the viral or the host gene expression. So regulation of either the viral expression or host expression, host gene expression, or by alteration of the host's immune response. So it can either regulate its own viral genes and how often they're expressed. So either it can just skip it altogether, so it would be a silent provirus or a silent persistent infection until it doesn't become one. So an example of this is a latent infection, which we'll talk about next. But a latent infection. A latent infection is where the viruses are capable of remaining hidden or dormant inside of host cells. So this is what we're talking about with it being silent. So viruses capable of remaining hidden or dormant inside host cells. So some examples, or an example would be chickenpox. So chickenpox is varicella zoster virus, or also something like herpes simplex virus. These are different types of latent viruses, which means that they get inside of the cell. Once they get inside of the cell, they integrate their genetic information into the nucleus, into our genetic information. And then it hangs out there. It's dormant. It's hidden. And then it can stay as that dormant state. And it can either stay as a provirus. So let's say either as a provirus, or it can be as circular DNA outside of the chromosome. So it can be either. So it can be a provirus or circular DNA that's outside of the chromosome. Either way, it's kind of staying hidden or dormant. And then sometimes what happens is that it can stay hidden or dormant like chickenpox virus. But then when a person is under stress and their immune system is decreased, then that virus can start to show itself. It'll start to make copies of itself, and the body won't be able to fight it off as easily. And so, for example, when you have chickenpox and you see a recurrence of that later, it's called shingles. What that means is that you've been infected with chickenpox. The chickenpox has gone dormant inside of the body, and it kind of hangs out there, not really doing anything, stays in there until your defenses are down, basically, which means that your body is no longer able to fight off maybe the little bit that was in there. And it kind of takes over, and it actually causes a reaction in the body. When it causes a reaction in the body, then we call that shingles in this example in chickenpox. So that's a latent infection. Then we also have chronic persistent infections. So our latent infection and chronic infection. So in the chronic infection, this is when a disease with symptoms that are recurrent or persistent over a long time. So this is when we have a disease with symptoms that are recurrent or persistent over a long time. Chronic is a long time. So in this case, what we're talking about is something like HIV or hepatitis C. So hepatitis C virus and HIV are two examples. that can cause a chronic infection. And so we are going to take a closer look at HIV right now. But what that means is that the virus is in there, so either as a provirus or circular DNA outside the chromosome, but it can exist as the provirus, and then in the case of HIV, what we're talking about are the immune system cells are going to be making copies, or it's going to be replicating its own DNA. And as it's replicating its own DNA, it's also replicating the provirus DNA, or the viral DNA, which is the provirus. Also, it's going to be transcribing into RNA and translating into proteins that provirus over and over and over again. So as long as that immune system cell is alive, it's going to have that provirus in it. And since it has that provirus in it, it's going to be transcribing and translating and making viruses. Now, at the same time, it's replicating its DNA and making copies. So if it's replicating its DNA and making copies, it's making new cells, and then those new cells have the provirus in it as well. And then it's going to go through its life cycle, or its length of life, making copies, transcribing, and translating the virus again. So then what we have here is that we have, say it only infected a single cell, a single immune system cell. It's integrated the provirus. It's making copies of that, sending out more viruses. But then when that cell replicates itself, makes a new version of the cell, because it's past its lifetime, it dissolves apoptosis. Now we have a new cell, and it's going to be making that same virus over and over again. So we're constantly sending out new viruses in the body, and we just can't get rid of it because it's a chronic infection, and it has symptoms. So in this case, we have the symptoms that go along with it. They're typically low-level symptoms because we just kind of keep pumping out virus. And in general, our body can keep fighting off the virus. But that's just in general. So we still have the symptoms. It's just more low-level rather than, having to lay in bed. However, in this case, since our body is just constantly fighting off a virus that we are constantly making, it means that we have a compromised immune system. And then that means that if we have a compromised immune system, any other infection that comes along, any other bacteria, any other virus that comes along is going to take its toll on that body. All right, so let's take a closer look at HIV and using the image that we find in our text. All right, so here we are with the image from our text. And I want to go through this because HIV is a really important virus to understand. So you should know these steps and how complex this virus is and why it's so deadly, or can be deadly. So first of all, what we've already spoken about with our eukaryotic viruses, or viruses that infect eukaryotic cells or animal cells, is that we have our attachment stage. So up here we have HIV is fusing to the host cell surface. And it actually takes at least 2 receptors to do this. And this is important when we get to chapter 18. We're going to be talking about our receptors. But specifically here, our CD4 receptor is an important one. For you to understand that HIV attaches to the CD4 receptor. It also requires the CCR5 or the CXCR4 receptor as a co-receptor. So it actually kind of attaches to the CD4, attaches to the co-receptor. Both of those attaching, you can kind of see that that is happening down here. We have a co-receptor attaching to the receptor. Once it does that, that then is going to tell the cell that this is okay, and it should bring it into the cell. So HIV is a retrovirus, and it tricks the cell into bringing it into the cell. And keep in mind that what it's tricking, remember we talked about different cells that are infecting different organs or tissues. I kind of skipped over it earlier, actually, but it's called tissue tropism. And tissue tropism is when viruses infect only certain types of cells. So infects only certain types of cells. And this is important because HIV has tissue tropism, which means that it only infects immune system cells, and only immune system cells that have the CD4 receptor, which are only some types of immune cells. And we'll look at that again in chapter 18 at the end of the semester. So it is going to attach to our immune system cell. It's going to co-attach to or use the co-receptor, the CCR5, and then that tells the cell, okay, go ahead and bring this in. So in step 2 here, we have the HIV RNA, our reverse transcriptase, integrase, and other viral proteins are going to enter the host cell. And you can see that that's happening here. So if you think about our 2 ways of our viruses entering animal hosts. Which one is this? We had the two different types, or the two different ways, remember, is endocytosis and membrane fusion. So this is an example of membrane fusion, right? So we can see that the membrane here, this is an enveloped cell. HIV is an enveloped virus, not cell, virus. And that envelope is going to merge with the plasma membrane of this immune system cell. So this is an example of membrane fusion. So the membranes are going to fuse, and then you can see that the rest of the virus is going to enter the cell. So the cell, or the virus, goes into the cell. In step 3 here, our viral DNA is formed by reverse transcription, right? So remember, we brought in reverse transcriptase with the viral RNA. Remember, what kind of RNA is this? Positive single-strand RNA. Okay, so we have a positive single-strand RNA virus, which is what HIV is. We also have reverse transcriptase. And remember that reverse transcriptase reverse transcribes, meaning goes from RNA to DNA. So we start out with our single-stranded DNA. Then we're going to turn it into our double-stranded DNA. And you can see that that's happening here with reverse transcriptase. We've turned it into double-stranded DNA, which is what this is here, our viral DNA, double-stranded DNA. Now we're going to be using the enzyme integrase that we mentioned to integrate that into the host DNA. So in 4, the viral DNA is transported across the nucleus, so we're going through the pore here, and integrates itself into the host DNA. So we use our enzyme integrase to integrate it into the host DNA here. So the blue is the host DNA, and it's going to stick itself right inside there. So now that it's stuck itself right inside there, now this immune system cell is floating around in the body, being an immune system cell, but as it is going through transcription and translation, you can see here in step 5 that new viral RNA is used as genomic RNA, and it's used to make viral proteins. So here we can see that it's transcribing our viral DNA into viral RNA, which to our animal cells just looks like mRNA. So it's just making copies of itself here. It's just not copies, but it's making RNA of itself. This RNA tricks our body. It just thinks that it's typical mRNA. So then it's going to go through translation here, right? So it's going through translation to make the protein. So you can pretend that there's a ribosome here using what looks like mRNA, but is actually viral RNA, and then it's ending up making these proteins here. These proteins are viral proteins. So then it's going through, this is over here. Let's say this is what's going to make up other parts of the virus. And then all of these things are coming together in here, and then it's going to bud off, and then we have our mature virion here. So in step 6, our new viral RNA and proteins move to the cell surface, and an immature HIV forms, which means that inside here it's budding off, but then it hasn't quite pieced itself together inside of that membrane yet. Then it pieces itself together, and then that means that the virus is now mature, and then this is when protease releases the proteins that form the mature HIV. So then we have our mature virion, and then it's going to go float downstream, and it's going to enter a new immune system cell via that CDR4 receptor with the co-receptor CCR5. Again, the reason that this is so dangerous is because this cell, this host immune system cell, is floating around the body all the time, just living its life as an immune system cell. It's going through, it's transcribing, translating, doing what an immune system cell does. However, as it's doing that, it is constantly making new copies of virus, constantly releasing those copies of virus that are then constantly infecting new cells that are then creating proviruses that we can't get rid of. Then that's creating more proviruses, but then as this immune system cell is reaching the end of its life, then it's going through and it's making copies of itself. And as it makes copies of itself, it's going to be replicating its DNA. When it replicates its DNA, it's also replicating the viral DNA, that provirus in there. And so then the brand new immune system cell that's never been an immune system cell before is going to already have that virus in it. And then as soon as it starts doing its job, it's going to be making copies of that virus and sending those copies out. So this is then causing that chronic infection that we talked about, where we're constantly sending out viruses, our body is constantly being infected by viruses, and then we're constantly having symptoms of that virus. And then, of course, our body, our immune system is decreased. Not only is it decreased because it's constantly fighting a virus, but it's also decreased because the actual immune system cells are the ones that are infected. So we're going out and we are destroying our own immune system cells because they contain the virus. But then we're trying to fight the virus, which decreases our immune system cells. So HIV, again, is a very, very dangerous virus, as are all reverse transcriptase viruses or retroviruses. So you should understand HIV and how it works, and in general, how retroviruses work and what makes them so dangerous. You should also know the difference between a latent infection and a chronic infection, which are both different types of persistent infections. So then now what we're going to talk about is the life cycle of viruses with our plant hosts. All right, so when we're talking about plant hosts, we're just going to talk really quickly about it. They are more similar to animal host viruses because they are eukaryotic than they are to bacteriophages. But when we talk about viruses with plant hosts, we're talking about plant viruses, of course. And plant viruses, I'm just going to talk about them in very general terms. So they may be enveloped or not enveloped. So, or not, which remember is called a naked virus. So we see both types. They may have DNA or RNA. So again, DNA or RNA. They can have single-stranded or double-stranded DNA or RNA. However, I should say may, most of them are positive single-stranded RNA viruses. So most of them are positive single-stranded RNA viruses. Some have broad ranges, so they can infect a whole bunch of different types of plants, maybe within the same family, or not even within the same family, just plants in general. And some have narrow ranges. So narrow ranges meaning it can really only infect one type of plant. So like in the text, I think it was talking about the citrus genus. So just one type of genus. Most of them are transmitted by contact between plants. So most transmitted by contact between plants. So the plants actually hitting against each other and then transmitting that virus, or by mechanical vectors. So mechanical vectors for plants would be like fungi, nematodes, insects, like arthropods. So things that are going to be on plants, and then they can transmit them mechanically. Remember, that's not biologically. It's not like an arthropod is going to bite a plant and then go and bite another plant or inject a plant. But that's them landing on the plant, and then the virus gets on their feet or whatever, and then flies to a new plant and then lands on that new plant, and the virus gets on that new plant because it comes off of their feet or whatever we're talking about. So a mechanical vector. Plant viruses are biotrophic parasites. So a biotrophic parasite is a virus that can infect the plant without killing it. So viruses that infect without killing the host. So similar to what we've already talked about previously, the infection can be latent. And when we talk about latent, that's asymptomatic. So meaning that we don't see some cell death In this case, we don't see plant death because it's a biotrophic parasite, so it kind of hangs out in there, but it doesn't actually kill the plant. But it can be latent, meaning asymptomatic, so it's just hanging out within the cells and is transferred from cell to cell to cell. And in some cases, some infections can hang out within the roots of plants and over again. Or it can be lytic. And so this includes cell death. So when we're talking about cell death, we could see the death of some pieces or parts of the plant, not the entire organism, but we can see different areas of the plant that might be dying, and that would be due to a particular virus. So that's really all we're going to talk about when we talk about plant hosts for different types of viruses. And now what we're going to do is we're going to move into talking about the viral growth curve. So this is the end of 6.2 here. We're going to talk about the viral growth curve. So when we take a look first, when we talk about a bacterial growth curve, what we see is called a sigmoidal curve. Sigmoidal curve. And so what this means is if we graph it out and we have the number of bacteria on one side and time on the other side, what we see is something like this. So we see that we inoculate, so we have some bacteria here. This is an incubation time here, and then we see a slow increase until we kind of max out, and then we actually can see a sharp decline after that. Now, we see something a little bit different when we're talking about a viral growth curve. And this is because the difference is between how bacteria grow and how virus or viral particles, quote, grow. So when we talk about the growth curve, what we are talking about here in regards to virus is on the side over here, we have number of virions, right, new viruses. And at the bottom, we have time. And so what we see here is the first stage is the initial stage. This is when we have inoculation of the virus, and that causes infection. So inoculation, and this is going to cause infection. And this is the initial stage. So our initial stage here, we're going to have a high amount here because we're going to put a whole bunch of virions inside of a solution. So if we can kind of think of it in terms of having a test tube that has a solution here with bacteria, okay? So we have a certain volume here of the solution. We're going to add the virus or the viral particles, so in this case we'll say bacteriophage, and once we add it to the solution we're going to have a high number here of those bacteriophages, right? Because we just added it to the solution. If we take a part of the solution now and measure it, we're going to have a lot of virions. But those virions then are going to decrease because they're going to be getting inside of the bacteria. So then it's going to move down and stay really low for a little while. This is called the eclipse. Whoops. Eclipse. So this is the eclipse phase. So this first part here was inoculation. Then we have the eclipse phase. So in inoculation, right, we have the inoculum of virus is going to bind to the cells, right? So it binds to the cells, but it's still outside. So it's kind of floating around and is binding to the cells. In eclipse, what we see is that these virions are penetrating the cells, and so they're penetrating the cells, and so they are getting inside of the cells, so we see a low number of virions in the solution. They're in the cell, and then they're going to hang out there for a little while because they're hijacking the cell's machinery, they're taking it over, they're making copies of themselves, they're assembling those copies of themselves, and then we have what's called the burst. So we have a high number here. We increase. This is called the burst. And what's happening in the burst is when the virions are released. So when virions are released, as the cells are lysed. So what we see is we see an increase in the number of virions, and we see a decrease in the number of bacteria cells or cells. So we have a burst, and unlike in the bacterial growth curve where it's very slow, they make lots of copies and more copies and more copies and more and more and more. So we see it's actually exponential growth in a bacterial growth curve. But that is different than what we see in a viral growth curve because we see this huge increase immediately because we see this burst because it bursts quite literally outside of the cell. Then what we have up here is called the burst size. And the burst size is the number of virions per bacterium. So this is the number of virions that are released per bacterium. So we can see this huge jump that levels off. That's the burst size. We can also measure what's called the viral titer. So the viral titer is actually the number of virions per volume of medium. And so again, we're talking about, let's say over here, we're talking about the test tube, and then we can take a particular volume amount of medium. So we would use a vortex, stir this up, and then we would take a volume out of there and we would count the number of virions per that volume, and that would give us our viral titer. And we can see this happening with humans, for example. So they'll take a blood sample to figure out what's going on inside of a human, and then they can take a portion of that medium and they'll see the number of virions per volume medium, and that's your viral titer. And you can tell where you are at, perhaps, in that viral infection. The burst size is more specific to the lytic cycle with bacteriophage, where we're talking about the number of virions that are bursting out of a bacteria cell. So this is our viral growth curve. You should be familiar with the different portions of it. So inoculation, eclipse, burst, and burst size, and also what viral titer means, what that is. This is wrapping up the last portion of 6.2. You should understand our viral infections with plant hosts. You should understand HIV specifically, but also in general retroviruses. You should understand persistent infections, the difference between a latent infection and a chronic infection, and some examples. You should understand the different types of nucleic acids coming in with viruses, so whether it's a DNA or an RNA, whether they're double-stranded, single-stranded, and if it's positive or negative, and what that means. So you should know when you need to use the RDRP and what that is, or you should know, if we're talking about a retrovirus, when you need to use reverse transcriptase. And then you should understand the viral life cycle of an animal cell or of a virus infecting an animal cell. So all of these different steps, whether it is included or whether or not it's getting into the cell via endocytosis or membrane fusion, and what that means. So how that's related to a membrane, whether it's related to an envelope or a naked virus, and what that means for inside of the cell.